Good afternoon, and thank you for your attention today. My name is Simon Richardson, and I am the Reader in Membrane Trafficking and Drug Delivery at the School of Science. I'm joined by my colleague and co-presenter, Benedita Ferran, and we'll be talking about our research. Before we take you on this journey, I would like to briefly remind you of some of the fundamentals associated with molecular biology. Critical to understanding molecular biology is appreciating the way that information is encoded into DNA. Not unlike computer technology, biology is also governed by code. In this instance, it does not consist of transistors and zeros or ones, but four shapes. These are the nitrogenous bases adenine, thymine, cytosine, and guanine, or A, T, G, and C, within DNA. Information is encoded into these bases through the order in which they are presented at the ribosome during translation. With the advent of epigenetics, we have come to realize that the flow of information in cells is bidirectional. However, the central dogma of molecular biology still applies. For the DNA that lives in the nucleus to affect change in the cell, it must be first transcribed into message RNA. Mature mRNA then travels to the ribosome, and they typically live either in the cytosol or on the surface of the endoplasmic reticulum. And it's here that the information encoded in the mRNA is translated into protein. On the left, you can see DNA code consisting of A's, T's, C's, and G's, where ATG initiates translation and TGA ends it. In between is the code for the amino acid sequence that, when properly folded, gives rise to green fluorescent protein. Protein folding is critical as protein function is always dictated by shape. This system gives rise to not only GFP, which has revolutionized cell and molecular biology, but also everything in the biosphere, from humans to bats to viruses. It is of direct interest as we have the capacity to edit this code and manipulate the biology of organisms or viruses this code runs. Here is a representation of the tree of life depicting the level of DNA sequence similarity between species. This is a crude representation of the diversity we see in nature, but how is there so much variation, and how do species evolve? If you cast your mind back to DNA code, all of the diversity seen in the tree of life is encoded by A, T, C, and G. Recombination and mutation within this code then gives rise to genetic diversity. Natural selection is the mechanism by which specific combinations of genes are selected in favor of others. This is dependent upon the environment and inter- and intra-specific competition. This is illustrated by the evolution of the peppered moth. The peppered moth has a white peppered phenotype and a black peppered phenotype. Prior to the Industrial Revolution, the environment in Manchester, England was pristine, and the predominant colour within the peppered moth population was white. However, after the Industrial Revolution covered everything with carbon, a shift in the predominant colour of the moths was recorded, with the black colour becoming predominant. Because of the change in the moth's environment, the black colour provided better camouflage from predators. This resulted in the black moth surviving and its numbers growing. We now understand the principles of natural selection at the molecular level, and how information encoded in DNA is stored in a cell. Photo 51 shows the first picture detailing the crystalline structure of DNA by Rosalind Franklin in the early 1950s. Just below this, you can see a confocal micrograph of two squamous epithelial cells. Here, the nuclei are surrounded by a ring of stained membrane, and within the nucleus, the majority of the cell's DNA is partitioned. The DNA that encodes the human genome, although ever-changing, has been sequenced. You can visit the Wellcome Collection on the Euston Road to see this information stored in a $3 billion bookshelf. Here, each of the folders within the bookshelf is labelled with a number, which corresponds to the chromosome containing the code detailed within the folder. This also highlights how much our understanding of biology, evolution, and genomics has grown over the last 100 years. It also underscores its power. As species become extinct and new selection pressures emerge, we have the ability to direct evolution at the level of code. One selection pressure that has recently stopped the world in its tracks is SARS-CoV-2, the virus responsible for COVID-19. Previous to the current pandemic, there have been several notable viral outbreaks. These include the Spanish flu in the early 20th century, the HIV epidemic of the 80s, the recent Ebola outbreak, SARS, MERS, and Zika virus disease. Viruses are parasitic machines that infect and hijack cells in order to produce many more copies of themselves at the host's expense. There are many different types of virus, which vary in their morphology, biochemistry, lethality, and infectivity. COVID-19 is especially problematic as it is easily spread and there is a delay in the onset of symptoms relative to an individual's infectivity. SARS-CoV-2 is an encapsulated RNA virus that enters the cell via ACE2 receptor-mediated endocytosis. Fusion proteins embedded within the spike protein found protruding from the capsid give the virus its corona-like appearance, mediate viral RNA escape from the endocytic system to the cytosol, where the virus can replicate. Once in the cytosol, non-structural viral proteins assemble into a replicase transcriptase complex, or RTC, where the virus's RNA can replicate, protected from intracellular antiviral responses. In parallel, viral structural proteins S, E, and M are synthesized in the cytosol, inserted into the ER, and trafficked to the ERGIC. From here, the virus is assembled and transported to the cell surface where mature virions are secreted. Understanding the virus's pathophysiology is the first step in developing a treatment. Several components of the virus can be targeted to arrest its replication. Here, a small sample of ongoing research, which includes repurposing small molecules and developing targeted biologics, is described. Biologics include neutralizing antibodies derived from people who have recovered from the disease, siRNA, and vaccines. These are all potential treatments. However, establishing their efficacy, development, and approval will take time. Of particular interest to us is the Vir Biotech siRNA program, which is attempting to develop an siRNA treatment to target this particular virus. This strategy is potentially adaptable and able to target any virus. This begs the question, what is siRNA? Humans have developed technology that can directly interact with genes or RNA in a sequence-specific way. This takes the form of gene editing using CRISPR-Cas9, for example. In the example shown, changing a specific base pair alters the 66th amino acid of GFP, changing it to blue fluorescent protein. We can also silence code using siRNA and antisense technology. Gene silencing using siRNA requires the delivery of a short piece of double-stranded RNA to the cell cytosol. Here, the RNA is processed by the dicer complex, and as a result, short interfering or siRNA is produced. The siRNA produced recruit a protein complex called RISC, which mediates the destruction of mRNA and inhibits gene expression in a gene-specific way. To date, 2 FDA and EMA approved medicines based upon siRNA technology have been approved. However, their use is challenging on account of their limited bioavailability. Bioavailability, that is, getting the drug to the right part of the target cell in the correct tissue, is limited by the labile nature of siRNA and the compartmentalization of the body at both the gross and cellular level. The body is composed of sheets folded into tubes which serve the purpose of compartmentalization. At the gross and cellular level, compartmentalization enables mutually exclusive processes like anabolism and catabolism, i.e. the creation and destruction of material respectively, to occur simultaneously in a regulated fashion. The endocytic pathway is one of the ways that compartmentalization occurs inside a cell. This serves to separate the outside of the cell, labeled A in the diagram, from the cytosol, B. In the instance of siRNA and ASO drugs, the target compartment is B. Confocal micrograph shows red-labeled protein called BSA that has been fed to a cell in culture. It can be seen colocalizing to the endolysosome where it is digested. This is an example of the compartmentalization that limits the bioavailability of siRNA and ASOs. As similar to BSA, they cannot diffuse across the endocytic membrane. Several protein toxins have evolved to be able to escape from the endolysosome, and a well-known example is ricin toxin. Ricin has a warhead, the A chain, and a rocket motor, the B chain. The rocket motor gets the A chain out of the endolysosome and into the cytosol across the membrane that would normally direct it to the cell's stomach and destruction. This is exemplified by the data shown in the bottom right of the slide, as when the rocket motor, the B chain, was removed, a 3 to 5 log decrease in toxicity was recorded. Here, we have used this rocket motor to deliver drugs like antisense oligonucleotides and siRNA to their target compartment, the cytosol. We tried with ricin, it was a disaster. We tried with shigatoxin, and it was also a disaster. We eventually tried with attenuated anthrax toxin, and everything worked. Anthrax is a disease that kills both cattle and humans. It's caused by a bacterium, Bacillus anthracis, which produces a tripartite toxin known as anthrax toxin. Wild-type anthrax toxin consists of protective antigen of 83 kilodaltons, lethal factor, and edema factor. PA83 is cleaved to PA63, which forms a heptameric pore enabling the translocation of lethal factor and edema factor into the cytosol. To detoxify anthrax toxin, we removed edema factor completely. We used PA83 in its wild-type form, as on its own this has minimal toxicity. We did, however, remove domains 2 to 4 from lethal factor to attenuate it, and fused in frame an RNA binding domain from protein kinase R. This formed LFN-PKR. siRNA can bind to LFN-PKR, which can translocate across the PA pore into the cytosol. This results in low toxicity and very high efficiency cytosolic access for the siRNA, helping to alleviate one of the rate limits associated with its bioavailability. This strategy is a little bit like removing the warhead from an ICBM and replacing it with a capsule that allows humans to live within it. This strategy allowed humans to go to the moon. Isn't using bacterial toxins dangerous? Clostridium botulinum serotoxin A, the active ingredients in Botox, has an LD50 in humans of between 1.3 and 2.1 nanograms per kilogram by IV injection. This means about 2/10,000,000ths of a gram is lethal to a person, making it one of the most toxic proteins known to science. The market size for Botox is predicted to reach $7.4 billion by 2025. And is evidence of market acceptance of the commercial use of protein toxins. My laboratory discovered that disarmed anthrax toxin could increase the activity of siRNA by about 300% compared to naked siRNA. Little or no toxicity was recorded with the delivery vehicle, up to and above the dose we used for drug delivery. We published and patented this, and now refer to it as our direct delivery technology. We were very interested to know whether we were using similar pathways and systems to deliver siRNA to the cytosol as is used by wild-type anthrax. This was a question that intrigued two of my current PhD students, Hadir and Norhan, who have demonstrated that our direct delivery technology is sensitive to inhibition by warp mannin, by wheat germ agglutinin, and by hypertonic sucrose. This means that it is very likely that intraluminal vesicles are necessary for the high-efficiency delivery of siRNA using our direct delivery technology. ILVs act as an airlock for the delivery of lethal factor and edema factor and can be seen in the micrographs to the right. When ILVs are secreted from the cell, they are called exosomes. And with that, I'm now going to hand over to my colleague, Benedita.
Exosomes are naturally occurring extracellular vesicles which act as carriers of protein, DNA, or RNA. Exosomes are formed in the endosomal compartment of most cells and consist of intraluminal vesicles prior to their secretion. ILVs, intraluminal vesicles, are packaged up with cargo whilst in the multivesicular endosome, and upon fusion of this MVE with the secreting cell surface, the ILVs are released as exosomes. The exosomes can then travel short and long distances to deliver their cargo to recipient cells. Exosomes are essentially a naturally occurring delivery system as they do not trigger an immune response, can evade phagocytosis, grant access from one cell to the cytosol of another, and also protect their cargo, often labile materials, from destruction. So why aren't we using exosomes for the delivery of drugs like ASOs and siRNA? Exosomes are really difficult to load. To the right is a Nature paper characterizing the loading of exosomes using electroporation. This is destructive and results in the disorganization of the exosomes, which impacts upon their ability to backfuse and thus deliver their cargo. How our technology differs from this method is that we load drugs, i.e. protein, DNA, or RNA, in situ into the ILVs. We then induce their secretion as exosomes, isolate, characterize, and use them to treat fresh populations of cells. Here we have a few examples of how we characterize these exosomes. Exosomes are typically 60 to 200 nanometers, and we can see from the photon correlation spectroscopy analysis that the majority of the population of exosomes we have isolated is just below 100 nanometers in size. The electron micrograph sitting just next to the PCS further confirms the size of the exosomes, verifying that they are indeed exosomes. To then assay for activity, we use the transgenic cell line, HEC293SCO08. This cell line overexpresses beta-galactosidase, an enzyme which converts lactose analogues from colorless to a bright blue compound, enabling us to detect substrate conversion spectrophotometrically. This assay was developed in our lab and has been extensively characterized over the past 2 years. So we loaded our exosomes with ASOs targeting this transgene and monitored their effect on the expression of beta-galactosidase. About 50% reduction in expression was recorded. We then loaded siRNA targeting the same transgene into exosomes and assayed their effect over time. In this instance, over 75% reduction in expression was documented. This is important because it shows that we can load exosomes. It also shows that the exosomes retain their ability to backfuse with the recipient cell, and that the drugs they carry remain active. So we show that we can load exosomes with a wide range of deliverables, which are active when used to treat a fresh population of cells. These include siRNA, ASOs, diphtheria toxin HA, and CRISPR-Cas9. The confocal micrograph to the right shows exosomes loaded with Texas Red labeled SA-Cas9 using our loading methodology. The university patented this technology in 2018. This is evidently a platform technology as the range of disorders it can be used to target is vast. Given the current landscape, we decided to focus on our work with viruses for this talk. So I'm now going to tell you a little bit about what we did in collaboration with the Bundeswehr Institute in Munich, Germany. At this point, I'd really like to thank the Institute and especially Dr. Joachim Bugert, without whom the following experiments wouldn't have been possible. We decided to use siRNA to target Zika virus because, in our hands, it's about 50 times more active than antisense on a molecule-to-molecule basis of comparison. So I started looking through the literature, and although, to our knowledge, no one had ever used siRNA to target Zika in vitro, there were several in silico papers with predictive sequences. So I scanned these against all the complete Zika virus genomes available in GenBank. The phylogenetic tree to the left shows sequence conservation of siRNA Ferrin ZV1 and all Zika virus genomes. I did exactly the same thing for all the sequences listed in Table 1 and mapped these siRNAs to their corresponding targets in relation to the NIH reference map of Zika virus. You can see it in green below Table 1. To test the ability of these sequences to target the virus, we electroporated cells with siRNAs 1 to 5 in HU and HEC cells and monitored cell viability 4 days post-infection. All samples were normalized to a no-virus control, which was considered our 100% viability control. We see significant protection of HU7 cells in relation to maximum lethality. However, HEC293 cells are not easily infected with Zika, and therefore the baseline death, i.e. the control, is raised. Consequently, the difference between treated and non-treated isn't as evident, although statistically significant for Ferrin ZV1, 2, and 4. In parallel to the nucleofection studies, we used our disarmed anthrax-based delivery system to test the siRNA's ability to reduce Zika virus-induced toxicity. But this time, we tested a range of concentrations from 25 to 200 nanomolar. And what we documented across the board was that siRNA was able to significantly protect BHK cells from Zika virus-induced toxicity. Here we also tested a commonly used drug, sofosbuvir, which in this instance didn't have a significant impact on the cells. We used the methodology described previously to load exosomes from HeLa cells with siRNA 1 and 2, and exosomes from HEC cells with siRNA 1. Their ability to protect HU7 cells was recorded in A and B respectively. These studies are ongoing, and a series of controls will be performed, along with new iterations of the experiments, which will include a dose response and a toxicity study. RT-qPCR will be used to monitor the downregulation of specific viral genes, thus validating the effect seen when monitoring cell viability. Joachim has asked us to design siRNA sequences which might be able to stop SARS-CoV-2, the causative agent of COVID-19. We began the siRNA design process by generating a phylogenetic tree of all beta coronaviruses. SARS-CoV, the virus that caused the SARS epidemic in 2003, shared a 79.9% identity to SARS-CoV-2, the virus responsible for the COVID-19 pandemic. For this reason, I began scanning the available literature for siRNA sequences shown to be active against SARS-CoV. This literature in patent search identified 30 siRNA sequences targeting SARS-CoV. The siRNA's ability to stop viral reproduction was rated on a scale of 1 to 10. SARS-CoV-2 homologous sequences were identified. The level of sequence conservation between the homologs and all of the sequenced SARS-CoV-2 genomes was investigated and ranked on a scale of 1 to 5, based on similarity. The homologs were subsequently aligned with the predicted SARS-CoV-2 RNA secondary structure and ranked 1 to 5 based on target accessibility. The conservation of the siRNA SARS-CoV-2 homologs was evaluated against all of the sequenced SARS-CoV-2 genomes on GenBank. Sequence conservation was ranked 1 to 5. The algorithm for ranking siRNA candidates prior to testing in vitro is shown in Table 2. The top 5 sequences were selected and have been mapped on the circular map of SARS-CoV-2. BF2 was cancelled because of its high GC content. These have been ordered from Invitrogen, and experiments should begin as quickly as the siRNA arrives. Dr. Joachim Boogert will start out using nucleofection, and then we will test out both our delivery system's ability to deliver siRNA and protect cells from viral infection. Coronavirus's reproduction is complex. At least part of the viral reproductive process occurs within replication transcription complexes, RTCs. These are relatively disorganized membrane brown structures that are thought to originate from the ER. They're thought to be another way that the virus hides from intracellular antiviral systems such as the interferon response. An obvious question would be, will RTC limit the siRNA response directed against SARS-CoV-2? Given that the siRNA we are testing was active against SARS-CoV, which was used to generate the data you see, there is every reason to believe that they will be. We have taken this into account when selecting our siRNA sequences, and will be targeting RNA translated both inside and outside the RTC complex. We know that activity within this complex is dependent upon cytosolic factors, which also means that RISC complex access to the viral genome may also be possible, though this conjecture remains to be tested. If you've made it this far in this talk, then you definitely want to check out both of these videos. Juan Enriquez is a lab favorite. Thank you for dreaming with us. I hope you enjoyed it.